Okay, good morning, everybody, and you're very welcome to the second webinar in the Teagasc Research Insights Webinar series. This series is aimed to address the challenges and opportunities in the agri-food sector, and the webinars will outline Teagasc's research and innovation in these areas. The current webinar trilogy focuses on the importance of soil health for underpinning food production and other environmental services. In today's webinar, we will have talks from Dr. Fiona Brennan on soil health, soil biological health, from Dr. Karen Daly on soil chemical health, and Professor Owen Fenton on soil physical health. I'd just like to remind you, as always, that there will be questions and answers at the end of the webinar, and I ask you to type a concise question into the Q&A tab and to indicate which speaker you would like the question to be answered for.
Hello. My name is Lillian O'Sullivan. I am a research officer with Teagasc, the Agriculture and Food Development Authority of Ireland. I'm very pleased to welcome you here today to Johnstown Castle, which is home to the Environment, Soils and Land Use Department of Teagasc. Since its establishment here in 1945, Johnstown Castle has been at the heart of soil research for the sector. Our soil microbiology team utilise a range of molecular techniques and functional assays to understand microbial communities and their activity in agricultural soil. This includes their role in soil and plant health, mediating greenhouse gas emissions, nutrient cycling and carbon sequestration. Soil analytical chemistry is the backbone of our nutrient, trace element and carbon research at Johnstown Castle. And our expertise in soil biogeochemistry and nutrient cycling is supported by a wide range of classical and novel methods that help us to capture changes in nutrient dynamics across a range of scales and soil types. Physical soil structure is threatened by compaction from modern heavy machinery and intense livestock traffic. A range of techniques can be used to assess soil structure, with visual methods, some developed by Teagasc, proving successful in identifying damage and highlighting where prevention or remedial action is needed. Healthy soils are critical to sustain life on the planet. Quite simply, as the European Commission proposed mission in the area of soil health and food indicates, caring for soil, caring for life.
So, as I mentioned before, the first talk is going to be given by Fiona Brennan, Dr. Fiona Brennan. She's a research officer in Johnstown Castle. Her research focuses on improving our understanding between the link between soil biology and soil functions, such as nutrient cycling and greenhouse gas emissions. Over to you, Fiona.
Thanks very much, Karl, and good morning, everyone, and thanks for listening in. So this morning we're going to be talking about assessing soil health, how we determine whether soil is in good health or not, what aspects we need to consider in terms of making that determination, and how research is helping us better understand how soil functions and to be better able to provide advice for sustainable soil management. There we go. So as was discussed in the last seminar by all of the speakers, our soils are incredibly important, not just for agriculture, but also for broader society. They provide a range of essential functions that are critical for life on the planet. These include functions such as providing our food, our fibre, our fuel, cycling our nutrients, purifying our waters, providing a home for biodiversity, and a whole range of other functions. When we think about whether a soil is healthy or not, we can think of it in terms of, is the soil able to provide these essential functions? How is it functioning? And in general, a healthy soil is a functioning soil. Nearly every soil has the capacity to provide multiple functions simultaneously. The degree in which they can provide individual functions differs between soils. So what I mean there is you could have a soil that's very productive, very efficient at growing grass, but that soil might have a less capability, an inherent capability to sequester carbon. That means when we consider soil health, we can't expect the same functions to the same degree from all soils. So we need to consider that one size doesn't fit all. Functions will be different from different soils. And also, every soil is important. So a balance between the functions in how we assess soil health is critical. The functioning of a soil is dependent on the physical, chemical, and biological properties of the soil. And these three aspects of soil health determine how a soil functions. They're completely intertwined and strongly affected by each other. So the physical characteristics of the soil, they provide the structure for plants to grow, for a home for organisms. They also control pore space, how water and nutrients flows through the soil. The chemical aspect provides the building blocks for plants and organisms to grow. And the biological aspect equally influences the physical and chemical. The biology is the driver for the nutrient transformations, and it also provides engineering services and stability to the physical soil properties. So together, these determine soil health, and we're going to break down today these three components. I'm going to talk about the biology side, and my colleagues are going to follow afterwards with the chemical and physical aspects. So when we talk about soil life, it can be difficult to know where to start because soil houses a staggering abundance and diversity of organisms, each with their own niche in soil, ranging from the microscopic, your bacteria, your fungi, archaea, etc., up into the larger organisms, macrofauna such as earthworms that we're all familiar with. In fact, soil is amongst the most diverse habitats on the planet in terms of biodiversity. It houses over a quarter of the total biodiversity on the planet, making it a globally important reservoir of biodiversity. The organisms have different sizes, different shapes, different lifestyles, and operate on a range of different scales. The diversity of the organisms is reflective of the dynamic and diverse habitat which is below ground. Soils have a very pronounced spatial structure. They have gradients of nutrients, water, organic materials, and this provides these habitats from which the diversity is derived. As the local conditions change, be it through the environment, through management, the number, the diversity, and the function of these organisms will change. So, of the three cogs, the physical, chemical, and biological, the biology side is the least well understood. And this is particularly true with respect to the smaller soil organisms, the microbial communities, which make up the largest component in terms of diversity and abundance. Traditionally, we would have tried to extract these organisms from the soil to grow them on plates in artificial media in the lab, or alternatively, put them solutions of them under the microscope to see what we could see. Unfortunately, this is extremely limited, and in terms of the total diversity that we would be able to look at, we can only get a tiny fraction of the total community with these techniques. And this has largely constrained our understanding of soil biology. However, new approaches are really revolutionizing what we know about soil biology. And now, instead of extracting organisms from soil, we can directly look at these organisms in situ in the soil and extract DNA, RNA, or protein from the soils and perform a range of new techniques on them, be it quantitative PCR, be it next generation sequencing techniques or proteomics. And all these can be used to extract from the soil genetic information about the entire community. Using these in association with biostatistics, bioinformatics, and machine learning approaches, we can, for the first time, look at what the composition of these communities are in different soils, and further, how they're influenced along a gradient of the environment or the soil type. So it's an extremely exciting time for soil biology because literally what we know is changing on a daily basis. But what does this mean in terms of management and understanding soil health? Well, we are looking at a range of different management options and how these impact on the health of our soils. And one aspect we're particularly interested in is looking at whether above ground diversity affects below ground diversity. That is to say, if what the crops you have in your field impacts the function and composition of the microbial communities in the soil. So we've been looking, as part of one project, looking at comparing the soil biology in monocultures of grasses, in legumes and in herbs, and seeing how the biology of those compare with a pasture where the species are mixed in a whole range of different ratios. And this enables us to come up with something like this, where we can actually predict, using some novel statistical approaches, what numbers of bacteria will be in your system dependent on the composition of those plant communities. A bit like the same way as you would use a texture triangle, we can look and see how many bacteria we would expect given a certain composition of plant community. But that doesn't tell us much about function, which obviously is extremely important. So we can bring this a step further, and using the same molecular approaches, instead of looking for counting numbers of organisms, we can target specific genes that we know are important for function. So this is an example of a gene that's involved in the denitrification pathway, NERK. And by counting the number of genes, we can look at the genetic potential of that community to denitrify and predict what that will be based on the community composition of the grassland. We can also directly look at microbial function by measuring using assays such as N-mineralization or decomposition or potential denitrification, and measure how much of a certain process will happen under the conditions and under what plant communities. So to bring this all together, the soil chemical, physical and biological properties of soil are completely intertwined. And if we really want to understand how soils function, we need to look at these three aspects together. In terms of soil biology, it's still science in its infancy, but technological advances have absolutely transformed our capacity to understand these communities, and we're learning new information all of the time. We can use these new technologies to understand what species are where and what they're doing, and how these are influenced by the environment and the soil type. And further, to understand what the impact of management is. To a large extent, much of your soil properties are fixed, and you can't control your climate, but you can control how you manage your soil. And by understanding these systems better, we can better inform how to manage these soils for more sustainable soil systems. And with that, I'm going to finish up and pass back to Karl. Thank you very much.
Thank you, Fiona. That was really fascinating to see. I suppose we now have tools to better understand the biology of our soils and what that biology means in terms of soil functions. Fascinating stuff. So just to remind all the attendees, if you have any questions for any of the speakers, please type them into the Q&A tab as concisely as you can, and indicate which speaker you'd like to direct the question to. So now we're moving on to soil chemical quality, and I'd like to introduce Dr. Karen Daly, who's a Senior Research Officer in Johnstown Castle. Her research focuses on soil chemistry in both an agronomic and an environmental context, and the application of sensor technologies in agriculture. Over to you, Karen.
Thanks, Karl. Good morning, everyone. Everyone can see my screen, I guess? So I'm going to start talking about soil chemical quality now. Essentially, what I'm going to talk about is things that happen inside the soil matrix at a very, very small scale, at molecular scale almost. We're trying to make sense of this information at field and farm scale, because that's the scale that we operate at, and that's the scale at which we can make changes to our management of soils so that we can improve their health. And also to put soils into a landscape context, because we have soils across the farm and across our landscapes. They interact with water, drainage, overland flow. They feature as part of ditches that are landscape features on farms, and subsurface drainage when you have water flowing above ground and below ground. It's all interacting with the soil matrix that it comes across, and the chemistry can influence what happens in the water. So environmental losses are often influenced by what's happening in the soil in terms of the chemistry. But inside the soil matrix itself, the main chemical reactions and the main chemical influences that we're interested in is nutrient storage and soil acidity, carbon storage as well. And the features of the soil matrix that you're looking at here are some of the principal components of the soil matrix that influence how nutrients are stored, how soil acidity is controlled, and how carbon is stored. So sand, silt, and clay are the three particle sizes that where surface reactions can happen. And so that those soil physical elements are all part of the soil physical quality. So there's an interaction there between soil physical quality and chemistry. And the organic matter is the grey clouds drawn in there. That's the sticky substance that sticks it all together. So organic matter is what holds the soil, holds the particles in place, and gives the soil some structure as well. So the biology is very important there, and that also influences the chemistry. So in terms of the reactions on clay surfaces, if we think of clays as sheets of paper in the soil, and on top of the sheets of paper are reaction sites. And within these reaction sites, that's where we can store our nutrients and our trace elements. So it could be any nutrient or trace element X, and that could be a metal, or it could be a nutrient like phosphorus, nitrogen, carbon, trace elements like copper, selenium, sulfur, etc. And they get on clay surfaces which control trace elements that we can store and their availability. So this is again coming back to texture and physical quality. The more clays you have, the more surfaces you have for reactions and for storage to happen. And you have organic matter that sits in between the sheets of paper. The sticky substance that I referred to earlier, that's the grey clouds there. And again, that gives that structural component and the biological component. So when we talk about what is inside the soil that can control the chemistry, the types of clay, the clay mineralogy is very important. So that's the composition of clays. There's many different types of clays, and so the composition of the clays in soil will often dictate what types of chemistry happen in the soil. The texture class is very important as well. So some of those texture classifications that you might be familiar with, like clay loam or a sandy soil or a sandy clay loam, etc. They all refer to the different distribution and composition of percentage sand, silt, and clay, and they also influence not just the physical quality, but the chemical quality. Structure as well, as I mentioned earlier, between organic matter and physical quality, holds it all together and holds it all in place and provides a medium for chemistry to happen. We can't control a lot of these elements. They're inherent features of our soils and our soil types around the country, but some things we can control, like pH, and pH is very important when it comes to soil chemistry. If the environment is too acidic, soils can often lock up or fix nutrients that need a neutral background or a neutral environment in which to function properly, become available as well. And the biology as well is very pH dependent. So, but pH is something we can control. We can control that with management in terms of getting a test for lime requirements to see exactly where you sit on the pH scales, and if you need to optimize or amend the pH using a soil amendment like lime, etc. So back to the soil chemistry controls. Nutrient storage, as I mentioned, is probably one of the main agronomic functions that we like to think about when we think about soil chemistry. So there's a lot of different elements in soil. There's a huge multitude of elements, but we're always fairly concerned about elements that we can control the inputs and offtakes of. So for example, phosphorus. Some of it's available, some of it's held and reserved, and some of it's fixed. But the reaction that we're very interested in knowing about in terms of research, and then further down the line in terms of refining our agronomic advice, is that reaction between the available fractions and the fraction that's held in reserve. We want our soils to be able to store nutrients, but we also want them to become available when there's a crop demand for nutrients. So that back and forth between the available box and the available reserves is a very important reaction, and it's what we know as what we kind of call them the absorption processes. So it's the process by which we're adding a nutrient to soil. We want the soil to adsorb it, take it in, but hold it there in reserve for when we need it, and make it available when we want it or when the crop wants it. So those absorption processes, in theory, it means that we're adding a nutrient to a soil, and the soil mops it up or takes it in and physico-chemically binds it to the soil matrix. This varies a lot from soil to soil. So I have 3 different types of soil here on this graph, shown by the 3 different curves. So basically, the top curve is a soil that will mop up a lot of nutrient when it takes it in and bind it very tightly. And the bottom curve is an example of a soil that has a very poor capacity for nutrient retention and for nutrient binding. This is almost, if you think of it in terms of a Connect 4, the more sites you have, which is the little holes in the Connect 4, or if you think back to my sheets of paper on the clay surfaces, the more absorption capacity the soil has. The fewer sites the soil has, the poorer the retention on the soil. So that's just one way of trying to visualize this. And this varies spatially from soil type to soil type. It is controlled by the amount of clays, the types of clays, and also the chemical composition, if there's a lot of aluminium in the soil, if there's a lot of iron in the soil. So a lot of those soil type effects can influence those very important processes that influence agronomic nutrients and their availability. But how do we measure these things in the lab? So for years, we've been using classical methods that measure soil chemistry, soil nutrient status, nutrients such as P, K, magnesium, and also lime requirement. And these are some of the standard tests that we run. You can run them in commercial labs. We run them here in Johnstown on a research basis, and it involves taking a composite soil sample, prepping the soil, and doing an extraction with a reagent, or maybe you might digest the sample if you're looking for the total number of elements or the total amount of elements, like something like carbon. If you're looking for total carbon, you might have to digest and combust the soil. But if you're looking for available nutrients, you might just use an extractant or a reagent. These tests are tried and tested. They're fairly standard across the world. They do generate a lot of waste though, because we do use a lot of reagents. And there's always a time and a cost associated with these things. So one of the innovations we're looking at in Johnstown Castle is green chemistry. How can we improve our methods so that we're not generating as much chemical waste, but we're trying to get the same information from soil and possibly more information from fewer tests. So one of the innovations that we've looked at recently that seems to work quite well for elemental analysis is X-ray fluorescence. So this is essentially shining an X-ray beam onto a soil sample without using any chemical reagents. We're not extracting anything out. We're not making up an acid or an alkali cocktail and adding it into the soil. We're just prepping the soil as we would have done previously and putting it into a little sample cup that you see here. We shine an X-ray tube hits the sample, hits the surface of the sample, and the X-rays penetrate the sample. The elements in the sample fluoresce because X-rays cause fluorescence, and you might know this from X-rays that are used in medicine. The light will fluoresce from what's being beamed at, and a fluorescence comes back to the detector. The detector sends the information to a computer which plots a spectrograph. So essentially we have peaks, and we can assign an element to a peak. So that's one thing that we're doing. And another that we're looking at in terms of green chemistry is infrared. So it's another type of spectroscopy. It's another form of light. And this, in this instance here, it looks for how chemicals at molecular level, how they vibrate and how they change. So the infrared beam will hit a soil sample. The chemicals in the chemistry in the soil sample, at molecular level, they'll absorb that energy from the infrared light, and they'll start to bend and stretch. And it's those vibrational movements at molecular level that send a signal to a detector. And what we get from this form of spectroscopy is a fingerprint or a scan of the soil sample. So this is an example of an infrared spectra, a mid-infrared spectra, and we can see peaks. So there's peaks and troughs from left, as you look from left to right. But what we're interested in is finding the peaks that relate to things like clay minerals and sand. And if we add in organic matter, we can start to build models that tell us a little bit about the absorption processes. And this is just from one scan of the soil sample. So we haven't measured 4 different things to get the same information as you would do if you were in a lab. It's a single scan. And then if we look for carbonates, which are in this region of the spectrum, we can start to talk about lime requirement, and we can start to build models that predict lime requirement. So from a single scan of a soil sample, you can get information on multiple parameters that would have taken you a much longer time in the lab, generated a lot of chemical waste and used a lot of reagents, and probably cost a bit more than what this does. So this information gives you information on the absorption processes, the types of soil, high P fixing soils, low P fixing soils, and the lime requirement as well. If we do this at national scale, we can build a fingerprint library. So it's like a spectral library of soil types in Ireland. And this gives us an idea then of what we can see, the bigger picture. So not just zoning in on one soil sample, but being able to compare multiple different soil types by looking at their spectral fingerprints. And this information gives us an idea of how we can build spectral models to predict multiple soil parameters from a single scan or one spectral library that you only have to generate once, and you have all the information there. So just to sum up, a lot of the chemical reactions and a lot of the chemical influences, the influences on chemical processes in soil, all come from other reactions in soil or other elements in soil that control physical quality and biological quality. So I would always think in terms of soil health as being the sum of its parts, the biological, chemical, and physical quality. They all have to work hand in hand, as Fiona said, because it's a very interactive system and a very complex matrix. That's all I have to say on the topic today, other than that you can follow our updates on our research at Johnstown Castle in Teagasc on Twitter. Some of us disseminate some of our latest work and our projects and papers on Twitter and on ResearchGate, but you can also contact us on Teagasc too. So I'll hand you back to Karl, and thank you for listening.
Thank you, Karen. I'm exhausted looking at all those molecules rocking and twisting. I didn't realize they were so energetic, and really interesting cutting edge research that you're carrying out across the country, and we look forward to the culmination of your project on the Teagasc survey with the Geological Survey. So now I'd like to pass you on to Professor Owen Fenton, who is a Principal Research Officer. His research focuses on improving our understanding between soil physics, water quantity and water quality, including drainage and remediation technologies. Over to you, Owen.
Okay, thank you, Karl. So we've heard about soil physical biology and chemistry, and now we're going to move on to soil physical quality. Okay. A soil that has good soil physical quality is one that has good soil structure. Soil structure is the architecture of our soil. It helps crops stand upright in the soil, but it also prevents things like erosion and compaction. Now, we've had studies in Ireland, such as the Square project, which was funded by the Department of Agriculture, which looked at soil physical quality right around the country. The overall outcome of that was that over 70% of the soils were in very good soil physical quality, but of course there was room for improvement. I'll show you more of these results later on. Now, a good soil with good physical quality enables good crop growth, but it also allows water storage and water transport, and it allows water, air, and nutrients to propagate down to the roots and allows and promotes flora and fauna. But of course, not all of our systems are the same. Grassland and tillage systems are quite different. Let's look at the cartoon here. You can see that the vertical stress that propagates into the soil from a cow is very different to that of a machine. Actually, the cow's propagation stress or compaction range is very shallow, and for a machine it's quite different, it's quite deep. And this means that we need very different techniques to assess compaction or soil physical quality, a change in soil structure in these systems. Of course, poor soil physical quality leads to other things. It leads to environmental degradation in terms of gaseous emissions and in terms of losses to water, in terms of runoff. So let's look at this schematic. It's very, very obvious looking at good, moderate, and poor here. Soil structure is very, very different. This is taken from the JRC website, and it really, really indicates on the left there's a lovely crumb structure right down throughout the profile. But as you move across, things become blocky and degraded in nature. Let's look at a tillage soil. Again, on the left, very, very nice, no pan, or a pan is a layer really which prevents water and nutrients passing through it. And as you move across, again, that pan starts to appear in a moderate pan, and poor, it becomes quite evident and quite thick in nature. And this has implications when we look at things like yield. For example, in the poor pan presence, you can see that the root structure has been negatively affected, and it even starts to move laterally in the soil profile. Now, Teagasc has produced useful in-field and laboratory tools to assess soil structure for all soils. So what do you need an in-field procedure for? For example, to inform management. Something that would take about 20 minutes, you'd be prepared to repeat that over time in different fields and use that information to inform your day-to-day management on a farm. But other things like laboratory tools are also needed. These are more high-end soil physics. They take a lot of time and detail. There's a bit of modeling involved. And these would be more suited, I suppose, to national monitoring networks when we look at soil health into the future. But they're also very, very useful for very, very sensitive changes in soil physical quality. And I'll show you an example of that later on. So the first in-field technique. Here's Julia Bondi, who has a very, very good paper, which you can find out more information. This is a machine learning tool for grassland. So in other words, here you dig a soil profile to waist height. Here's Julia looking at that soil profile in detail and looking for visual indicators. The tool predicts soil horizon bulk density, and we all know bulk density changes over time. So therefore, it's a great indicator to look and track management on the site. The model initially took 11 visual indicators. Think about texture, structure type, humus, etc., but eventually broke it down for 7 visual indicators, which give an in situ harmonized bulk density value. This is very, very useful because it can be converted into something like a phone app, which means you have the lab in the field. While you're looking at the soil profile, you can determine not only these visual indicators, but also bulk density. Bulk density is very useful for other things as well. Think about carbon stocks, for example. Okay, now I'm going to move on to a visual field scale grassland management tool. Some people may be familiar with VES, or visual examination of soil structure, and that's a more generally used tool, particularly in the tillage sphere. We decided to look more closely to develop a tool for grassland sites. And what's different about grassland is this root mat. A root mat on top of the soil is like a sponge or a roof, which dampens the effect of compaction, for example. So we felt we needed to develop a bespoke method that takes into account this root mat. And in conjunction with University College Dublin, here's Jeremy Emmett Booth, who devised the scheme. He's demonstrating the method in the field. It's very, very easily learned, and it takes about 20 minutes to deploy. You basically take out a block of soil, you open it like a book, and then you examine the structure in that block. Jeremy has also produced the Soil Structure ABC booklet, and I would really encourage people to look at the link below, and it's freely available, and it shows the Grassfest method. It looks like this, okay? There's a lot of detail here, but all I want you to show or to indicate is that you start in the flow chart, you work your way through that block of soil, and it brings you to conclusion. SQ1 in that green zone is a good soil structure, and the red zone is a degraded SQ4 score. You combine that with your root score, and it gives you an overall score for your soil structure, which indicates a good or a bad soil physical structure. Now, going back to the national picture, the Square project conducted Grassfest all over the country in different agri-climatic zones, and you can see the results were very, very positive. If you look at the graph here, the vast majority of soils were in that SQ1 zone, with some soils above SQ1, 2, in fact only 3 were above SQ3. And these zones were atypical zones, we call them. So when Grassfest was deployed in areas like gateways, around water troughs, at the side of fields, or in areas where wheel rutting has occurred, degradation was evident. So overall picture is very good, but definitely room for improvement. Now let's move on to Irish tillage soils. The challenges here, I suppose, are as indicated by that vertical soil stress, the weight of machinery. You get a deeper impact in your soil. And in wetter climates like in Ireland, this is certainly challenging. Also, we need to defend organic matter in our soils. Organic matter really is the glue that binds that architecture together. And this is at risk in, I suppose, tillage soils. Here's a slide from Dermot Forrestal in Oak Park, who developed methods for tillage soils in connection with University College Dublin again. Here's the double spade method. So you can see in the diagram, the vest goes down to about 20 centimeters. The double spade, as indicated, it's double that depth. So you dig out your extract down to about 20 centimeters. You take that out and then you dig down again to 40 centimeters. The results here indicate that there's this transition zone layer, which the double spade really, really picks up well on. And it was more sensitive than quantitative methods. So in other words, the double spade method gives you more information about your soil profile than the normal vest method. And it's very, very useful. For example, you can assess structure and then avoid crop yield losses. So if this is done before crops are planted, you can pick up areas that will be prone to compaction, alleviate those areas, and then you're not at risk at losing yields. Dermot also inspected headlands and machine traffic impacts. And if you look at the pockmarks here on this photo, they're the double spade methods being deployed by the PhD student here in the field. They were conducted at the transition zone, the turning zone, where the machinery turns in the field, and right out to the edge zone. 40 sites were surveyed, and you can see here that most damage was caused in the turning zone, where machinery turns and heads back in the opposite direction. But soil effects were not responsible for all headland yield losses. So this turning zone was very, very interesting. And it was another experiment focused on this across 35 farms. Results showed that 30 to 65% of headlands had compacted soils, and it was the weight, tires, and pressure impacts that created this soil stress. So, of course, there's scope to reduce damage by altering machine specifications and by controlling traffic. Now I'm going to leave you really with this last bit. This is the high-end soil physics and laboratory option for all soils. You can see here that Matthias here is out in the field. He's taking an intact soil core. He brings that back and he develops what's called a soil water retention curve. That's the relationship between soil, water, and the plant. It's actually that suction that goes in and out of a root. And he then develops these very, very high resolution soil water retention curves. And you can do lots and lots of things with this data and really, really find sensitive changes in soil physical quality in any soil. In fact, Matthias was interested in the crossover between soil physics and soil biology. And he looked at how earthworms travel in soils to a food source like a cow dung pat, to it and away from it as the dung pat degraded. And he was able to track, using these sensitive methods, the changes in soil structure and soil physical quality due to even earthworm movement. So now we can not only look at changes due to machinery, but also to earthworms and everything in between. These kinds of tools will be really useful for national monitoring networks into the future. So to finish, and in summary, the state of Ireland's soil physical quality is very good, but problem areas exist in these atypical areas. There are in-field quick tools to inform management in real time, but remember, different tools are applicable for grassland and tillage sites. So these should be learned, they're easy to deploy, and they can be reused over time, and they inform management. Laboratory methods, which are the most sensitive, could be used for national monitoring programs. And with that, thank you very much, and back over to you, Karl.
Okay, thank you very much, Owen, for a very insightful talk. So again, that's the end of the 3 webinars on the biological, chemical, and physical health of soil, and some of the ongoing research and innovation within Johnstown Castle. So we're going to move into questions and answers now, and I'd like just to remind people again, type your questions into the Q&A tab and indicate the speaker that you would like to address that question. But then in terms of speakers, if there is a question that you would also like to contribute to, maybe speak after the primary speaker. So for this, I've got some assistance from David Wall to help me sort through all of the questions that we've received. So starting off, I suppose, Fiona, there's a question here for you, but other speakers might want to comment on as well. And that's what management options are available to farmers to improve the health of their soils.
Yeah, so that's a really good question. And actually, there's a whole range of things that farmers can do to enhance their soil biology. I'd say first and foremost, following on from what Owen has said, avoiding physical damage to your soil is one of the most important things you can do for your soil biology. So keeping a living root in the ground, and also avoiding trafficking, animal traffic or machine traffic when soil conditions are unsuitable, because these can cause either erosion, where it's total loss of the carbon-rich topsoil, or can cause compaction, where you're literally squeezing out the pore space, and this is where the biology lives. So avoiding physical damage. And I would think Owen's suggestion of farmers, if they can, getting out every now and again and checking the physical quality of their soil with those simple methods, Grassfest or the double spade method. And they can even combine this with some biological assessments, such as earthworm counts. It can be very useful to see what areas, if you have problem areas on your farm, and to be able to address them. The other things I'd suggest in terms of soil biology is, if you can, try and diversify your crops as much as possible. Monocultures above ground equals monocultures below ground. So it reduces the diversity of the environment. The root architecture becomes uniform. And in terms of carbon inputs, it's much less than a mixed community. So in whatever way you can incorporate that into your system, if it's a cropping system, maybe you could have crop rotations or cover crops, or intercropping in some countries they have. In grassland systems, try to get as much diversity as you can into your swards. That could really help in terms of your soil biology. The other things you might think about in soil biology is to try and get as much organic matter into your soils. This is particularly true for tillage soils, where carbon can be lost just from the disturbance of the soil, but also in grassland systems where you have continuous silage without return of organic matter. So trying to get organic matter into soils, it really is the glue that holds everything together, and it's the food for the biology. So get in your farmyard manures, your slurries, cover crops will also help in terms of carbon being returned to the soil, and any form of carbon that you can get in, really, crop stubbles, etc. And the final bit of advice is try and keep your fertility balanced. Don't over apply fertilizer. It favors proliferation of certain organisms and reduces the overall diversity and abundance. And trying to keep those fertilizers balanced. So don't run down your pea, for example. The organisms need all of those nutrients to keep functioning. So that would be my main recommendations.
Thanks, Fiona, and I think you probably covered what the other panelists might have said as well. David, I have David Wall helping me here sit through the questions. So David, anything jumping out on the questions from you?
Question coming through for Karen there. It's a two-part. First, somebody is a little bit concerned about, you know, X-rays and I suppose a little bit of human health, or the person in the lab, but the more practical side of the question is, can these new methods inform between total elements in the soil and the available elements that we expect for our plants?
Okay, thanks for the questions. On the safety aspect, X-ray fluorescence yes, it does use X-rays, and if you want to go down that road and analytically, you'll need a license from the RPI. So our lab has a license, and that's where we store the equipment, and we have all of the safety protocols around using X-rays in chemistry. The box that it's sealed in, you know, the beam is inside a lead box. You can't open the box if the beam is on. There's a lot of safety controls around it, and we've all been certified users of X-rays. So you know, you can't use it until you've been licensed, certified, trained, the whole lot. But so there isn't really a health risk there. In terms of the application, what I will say about, I talked about two different types of green chemistry. The XRF does, when you look at when you point the laser at a solid sample, it will look for the total elemental amount. So it'll look for all the elements on the periodic table except the lighter elements like carbon and nitrogen, unfortunately. So, but it can look at total phosphorus and total potassium and total iron and aluminium, etc., etc. So that's if you want, and this is something we are working on, if you want to only look at a certain fraction, you can take that off first. And so there's a couple of different methods. You can diffuse off. So instead of using reagents, you might be able to take out a soluble fraction in water, and you can put that into the XRF. So XRF can measure solids, liquids, and kind of powders and gels. You can also diffuse your available nutrients onto a gel disc and then put the gel disc into the XRF. So there are ways and means. They are new methods, so it gets around the use of chemical reagents. As long as you find a medium on which to diffuse your available nutrient, then you can show it to the XRF or point it at the laser, and it'll measure what's there. In terms of the infrared and available nutrients, what we're trying to see there are the processes that control nutrient availability, not necessarily the quantities of available nutrients. So those absorption processes, those other features like acidity, the amount of clays, the organic matter. So we're looking at not so much the exact amount of a nutrient, like for example phosphorus, but the processes that control it, so that we can say something about different soil types and their behaviors, so that we can start to think about soil type specific management advice in terms of fertilizer applications. So the IR will give you information about the processes that go on in the background, rather than the actual quantities itself. And the XRF will give you totals, but there are ways and means of getting availables as well. You just got to tweak the method a bit. I hope that answers that.
Thank you, Karen. There's another question coming through here in relation for Owen Fenton in relation to the tillage image that you showed. And the question is around what causes the pan presence in those soils. And then I suppose, look, another question also came in around clay content. I don't know if you know what sort of clays that we have, but there was a question around do we have soils with mainly non-expanding clays, ileites, versus montmorillonite. So I don't know if you have an answer to that. It's very specific, but what causes the pan to form within those tillage soils?
Okay, very good question. So I suppose a pan is like a layer that's very, it's almost like really compacted layer. It's very tight. A lot of people think that pan layers can be very thick, but actually they can be even like a pencil line in some cases, and they can go right around a soil profile when you dig it. If you think about it, a vertical stress creates compaction down into the soil, and it creates a lower permeability layer. And then as nutrients, for example, iron could be washed down through the profile, and it starts to accrete or form this layer. And that's why sometimes they're often a kind of a red hue. It's just that mineral layer building up. Pardon me. And if you think about it, it then acts like a barrier, you know, so you've less water storage, less water transport through that layer. And then if you have roots that want to go down through that layer, they cannot, and that has yield problems. So it's a natural feature, but it really is caused by management, alleviation of of a ban is quite controversial in a way. A lot of people are advocates of subsoiling, but always think about subsoiling. You need very, very long, prolonged dry conditions for subsoiling to be effective. And often in countries like Ireland, where windows of opportunity for those long periods aren't very, very valid. So, I suppose prevention is always better than cure. I really would repeat that. And there is always a health warning when it comes to alleviation using subsoiling techniques. The second part, I think, was about expanding clays, illite versus montemurlonite. I'm going to be honest here, and I wouldn't know that in very, very detail. In our soils, I would know, even in our heaviest soils, for example, across the heavy soils program, the amount of clay we would have would be really in the highest would be in the 30s, 30%. Within the Irish Soil Information System, I don't think there's many soils that would go above 40%. Just to give you an idea, it's very much our heavy soils really are confounded by our poor weather or rainfall in the west of Ireland in particular, and we have trafficability issues and getting out under conditions where the field capacity, is about less than 60%. So we have to be mindful of when we're using our soils to prevent compaction to then which leads to problems for gaseous emissions, runoff to water, and all of the other things that were mentioned here this morning.
Okay. Look, thank you very much, Owen. David, any other questions jumping out?
Yeah, there's one on the practical side. I think for Fiona and Karen, maybe you want to answer. What practical applications can we test for? So what's practical to test for other than, we'll say, standard PK, lime, etc., that might inform or assist future management?
Well, I can start off. Yeah, I can start off. The first thing that would come to mind, beyond the standard, suites of PK lime requirement, percentage organic matter is a really good indicator of soil health, and it's also a good management tool as well, because it gives you an idea of where you are on the spectrum of organic matter. And Irish soils have a lot of organic matter. Like, we're not degraded soils that you would see in a more tropical climate. We do have soils that, they start off maybe at around 4 or 5% organic matter, and that can go up to 90%. And there's a lot of in between. Like, the range is very, very wide, because there's a lot of wet ground, there's a lot of heavy ground, there's a lot of peaty soils and soils that are somewhere in between peaty soils and mineral soils. And they all behave very differently in terms of nutrients and nutrient retention. And so their biology is going to be different as well. So if you were to pick one other kind of, management indicator that could give you a snapshot of where you are on the organic matter spectrum in terms of how you might manage your nutrients, but also in terms of what the soil health might be like, I would go for that one.
Okay. Fiona, would you like to add briefly?
Fiona, do you have anything else to say?
So I would agree wholeheartedly with what Karen has said. Organic matter is actually a great thing to look at. But I think looking at soils, you really have to look at the broader context. And you can get an awful lot from those visual assessments. You can look at your nutrients. But from the different elements, you can really tell how healthy your soil is or not. But organic matter is a fantastic one to look at. And even as well, if you want to bring it a step further, looking just not at the components within the soil, but also the processes. So I don't know if some people will have done this soil your undies, where you're looking at decomposition processes, or ones that look at respiration. All can give you a good indication of how your soil is functioning and what its health is. Thank you.
Okay, thanks, Fiona. And that does line up the webinar in 2 weeks' time, which is going to focus on soil carbon. So tune in for that. Fiona, there's another question here in relation to some of the results that you were showing around diverse swards.
Yeah.
Do they give a more diverse microbial community, and have you found any links to between that community and denitrification or other soil functions?
So international research would suggest that we'll get a more diverse community with diverse above ground. That work I showed is actually still ongoing. We have the initial results on the function side, and it does look like depending on your plant community, you get different functions, but it changes depending on what function you're looking at and what plant species you're looking at. And also, we'll be looking at the community composition. So hopefully by Christmas, we'll be able to give that answer to that question on our own experimental trials.
Okay, thanks, Fiona. Owen, there's one here for you around, I suppose, what innovative soil physics facilities do we have? How can you, or how would you to assist in the soil, I suppose, physical health assessment?
Yeah, okay. I suppose like Karen, we're moving from traditional techniques and trying to, I suppose, bring innovation into the soil physics space. While I suppose validating our results for the time being with those traditional methods. Things like creation of soil water retention curves where you previously would have used sandbat, pressure plates, etc. We still are using those, but we've really moved into the high resolution space where we're using things like evaporation methods, centrifuge methods, and we're able, with using R code, to really produce high resolution soil water retention curves. That leads to way more capacity in terms of indicators into the future. We're working with colleagues in Brazil at the moment, and we're looking at our Mindo and Vendroche method, which really looks at the area under the curve, and it's extremely sensitive. I think it's more sensitive really than anything else out there. So this could be used particularly to look at soil biology and soil physics relationships into the future. We have a full soil physics setup now in Johnstown. We can look at texture. We can look at field and laboratory techniques in that area. So yeah, I think we're really well set up for the future. Yeah, and we're open for collaboration in the soil physics area.
Thanks, Owen. Look, and a final question, and look, apologies to those who haven't got an answer to their question, but we will put up some written answers to those. So it's a question for all of you, and it's not going to be, maybe a quick answer if you can. Is there any evidence that soils under organic farming or regenerative farming practices have healthier soils?
And maybe I take that first from the biological perspective. Yes, they do seem to, from international evidence, have a more healthy biology because they tend to have more organic matter, more diverse species, and less fertilizer being applied. So that would be the general, I suppose, perspective on it.
Okay, look, I might stop it there because it's 10:30 and we've been going for an hour now. I'd like to thank everybody that has attended today's webinar. And again, you're welcome to follow up with the researchers, both Karen, Owen, Fiona, with any specific questions that you have. Their presentations will be put up online. So go to the webinar series webpage for more information and links to the relevant documents or papers or other websites that the speakers have. The next presentation or the next webinar is in 2 weeks' time, and that's going to be taking a closer look at the importance of soil for removing and storing carbon from the atmosphere. We've heard all the speakers talking about the importance of organic matter, and primarily that is carbon. So tune in for that in 2 weeks' time on the 26th of September. And finally, there will be a feedback survey when the webinar is over, and we will greatly appreciate if you could give us your feedback and comments to help us improve the webinars for the future. So Owen, Karen and Fiona, I'd like to thank you very much for your insightful presentations, and thank all the audience for their interesting questions and their... Thank you.
Thanks, Karl. No problem.
Bye.